This screencast will deal with control of microorganisms, and basically we're going to look at antibiotics and disinfectants. When we deal with control of microorganisms, there are a lot of reasons we have to deal with this because microorganisms are everywhere and they do all sorts of things. Our challenges, so to speak, include trying to prevent contamination of inanimate objects when we eat or use things in the medical area. We've got all these materials that need to be cleaned and need to have microorganisms controlled on them. Whereas our second challenge really comes down to how do we kill and get rid of things that are infecting living organisms without destroying the living organism. When we look at principles of control, there are a lot of different terms that we deal with, which include things like aseptic, and aseptic is any environment or procedure that is free of contamination by microbes, which means that you may have a specific microbe there, but it's not being contaminated by other ones. Decontamination says we're looking at treatments to reduce pathogens to a level that is considered to be safe. Here we're looking specifically at pathogens, so to speak. So when we decontaminate something, we want to bring the levels down so that it's safe for virtually anybody. Degerming is the removal of microorganisms from a surface by scrubbing, and when we do degerming, that's really what a physician does when they scrub their hands. They go out and they remove all sorts of microorganisms and dirt and everything else, and they can actually do it quite well just by scrubbing and taking the material off that way. We have sanitization, and sanitization is a process of disinfecting places and utensils used by the public to substantially reduce the number of pathogenic microbes. Sanitization is not really a level of control, but it meets public health standards. So they say, public health standards say that you shouldn't have any E. coli in the food. Well, you can have other things in there, but it doesn't mean that it's sterile, but it just means that it's been sanitized. We have sterilization, and sterilization says we're going to remove or destroy all microorganisms, including endospores and viruses. So when you're talking about sterilization, we mean the environment is sterile. There are no longer any kind of microorganisms. Then we have disinfection, and disinfection says we use physical or chemical agents to inhibit or destroy most microorganisms. When we deal with disinfection, we use a disinfectant if it's an inanimate object or surface. So in your kitchen you use a disinfectant on the cutting board, on the countertop, on the table. We also have an antiseptic, and an antisepsis is when we use an antiseptic, which is a kind of disinfectant on skin or other living tissue. So when you've got a cut on your arm and you use a material to disinfect it, that is really an antiseptic that you're using. If we look at it, we think that we could use hydrogen peroxide to clean a tabletop. We could use hydrogen peroxide to clean tissue. If you did it on a tabletop, it is a disinfectant. If you use it on tissue, it is an antiseptic. When we deal with these, we often look at antibiotics because antibiotics are extremely important to us. And antibiotics are basically antimicrobial chemicals produced naturally by microorganisms. There are many different types of antibiotics. The first one was discovered by Fleming. What he had was a culture plate that got contaminated by a microorganism, just as you see here. This is a blood agar plate with Staph aureus. You can see the penicillium growing on it, and the penicillium is inhibiting the growth of the staph. By doing that, he started saying, wait a second, we've got some interesting material here that we need to go out and we need to further understand. One of the ways that we scientifically deal with these materials is through what we call the Kirby-Bauer Test. And the Kirby-Bauer Test, it really determines the susceptibility of a microorganism to a specific antibiotic. We use what we call a zone of inhibition, and we can compare that with a zone diameter interpretive table so that we can do these tests virtually anywhere in the world on different microorganisms and see how specific things work. The size of the zone of inhibition depends on the rate of diffusion and interaction with the microorganism. It also looks at the number of microorganisms that are initially present, how rapidly the microorganism grows, and how susceptible the microorganism is to the chemical. So if something diffuses very quickly, you might have a very large diameter zone. If it diffuses very slowly, you would have a small diameter. So there are a lot of different things that we look at in here. When we look at the Kirby-Bauer test, it is a standardized tool. It is standardized in the amount of material you put in the culture plate. It is standardized by bacterial concentrations and things. So when we look at it, it's a standardized tool. We use it for measuring the effectiveness of antimicrobial chemicals against pathogenic microorganisms. It is standardized to ensure reliable results, to make sure that everything works exactly the way it should. When we deal with these, we look at the zone of diameter in millimeters. The way this works, and we've got a video on how to set this up. This shows you an antibiotic dispenser. You can see on the left-hand side the dispenser with the various tubes in it. Each tube has little discs. Each disc has a letter or letters on it that indicate the antibiotic. It also has a number that indicates the concentration. The way these work is they're all very similar. The antibiotic dispenser, basically as you press on it, you cause a motion that will knock out a antibiotic disc onto the culture media, and once you do that, the discs will be held in place, and you can then look at the zone of inhibition that is produced. You can see the discs down in the lower right hand corner. This is a plate where you're looking at it several days after it was inoculated. As you can see, various zones of inhibition. The way you measure these zones of inhibition is you look at it. If we go over here and we look at the P, we can see the P10, there is no zone. We look at the TE30 down here, we've got a little bit larger zone. The S down here has got a little bit larger zone still, and the E over here has a much larger zone. And what we do is we measure the diameter of the zone of inhibition in millimeters. Once we've done that, we can go and we can look at the material on the zone diameter interpretive table. This indicates a couple of different kinds of zones you've got, and again we said that the letters stand for the antibiotic, the number stands for the concentration of that particular antibiotic, and we always look at the diameter in millimeters. Now you just have to be careful that when you record these that you actually record them in millimeters. We can then come to the zone diameter interpretive table, which is this. On this you can see that first column shows you the symbol of the antibiotic. The next one gives you the antibiotic name. Then you've got the concentration. Then it gives you numbers. So let's pick one out. Let's say we were dealing with ampicillin. It says ampicillin is AM. Concentration is 10 micrograms. It says if we're looking at gram-negative bacteria and enterococci and we get a number of 13 millimeters or less, then that bacterium is considered to be resistant to the antibiotic. If it is 17 millimeters or more, then we're looking at the organism being susceptible to the antibiotic. But look, down below we've also got ampicillin. It says here we're looking at staph and penicillin G susceptible organisms. A resistant value is 28 or less. So you can see this is much less than the 13 above. In order to be able to consider the organism to be considered susceptible, it has to be at 29 or more. So there are many different ways that this table becomes interesting. You have to go out and find your specific symbol in the first column. Then you look at the concentration, and all these concentrations are standardized. Then you come out and you interpret it with your diameter of your zone of inhibition, and that will give you a reading as to whether or not the organism you have is resistant, intermediate, or sensitive to the antibiotic. We can also do the same sort of thing with disinfectants or antiseptics, and we do this in class so that you can get an idea of what's going on here. And you can see with these we've got some different size zones of inhibition. You can measure these, but there is no standardized chart to use with these particular ones. So again, control, we're looking at trying to deal with these microorganisms, how to get rid of them, how to control them, how we can live our lives without them damaging us at different times.